Hello, everyone, and welcome to today's webinar. I'm Charlene Lancaster, Assistant Science Editor for The Scientist, and I'll be moderating our discussion. Today, our speaker, Evelyn Lattmann, will present an artificial intelligence-based microscopy approach that enabled her to morphologically classify melanoma cells without fluorescent labels. Before we begin, we have a short presentation from Deepcell.
Hello, and thanks so much for being here to learn a bit about the Deepcell and how it's being used to study cancer. My name is Michelle Balakrishnan, and I'm a Senior Product Marketing Manager at Deepcell. I'm going to give you a little bit of a background on the technology underlying the REM-I platform, and then I'll turn it over to Evelyn to share her compelling research. So, in the age of single-cell biology, there are scenarios where trying to isolate or enrich a specific cell population is not possible because there are no direct markers available for that population. Imagine using single-cell RNA-seq. You identify a rare cell population that is resistant to a drug or is disease-specific, and you want to be able to identify these cells for further analysis, but there are no markers available to isolate and characterize these cells. What do you do? Our viewpoint at Deepcell is that morphology itself, which is indicative of both phenotype and function, as demonstrated by many publications, is a modality that can not only add additional context for understanding single-cell biology, but also provide a method for identification and sorting of cells like those in the scenario I just gave. However, cell morphology is highly dynamic and complex, and analyzing morphology by eye can only tell us so much. There are obvious morphological differences, like the top example here, showing a mesenchymal cell and a melanocytic cell. We can easily see the differences between those two cells. But look at the bottom example. The drug-sensitive versus drug-resistant cells aren't so easy to distinguish by eye. So, in order to leverage morphology as a modality for identifying differences between cells, we need to turn morphology into a more quantitative, high-dimensional, unbiased method. Turning cell morphology into a quantitative, high-dimensional ohm, the morpholome, brings it in line with other traditional omic methods to round out a more comprehensive multi-omic analysis and more fully characterize the biology of single cells. At Deepcell, our approach to morpholomics is to provide single-cell, high-dimensional, unbounded morphology analysis via the REM-I platform. The REM-I provides high-resolution imaging of single cells, high-dimensional analysis of morphology, and label-free sorting in one platform. Now, let's dive into each of these components a little bit more. The REM-I benchtop instrument is where samples are loaded onto an imaging or sorting chip, and while in flow, high-resolution bright field images are taken of each cell in a sample. The real-time analysis provided by the AI means that the workflow is fast and able to sort viable cells into 6 different collection wells. This completely label-free workflow is gentle and maintains cell viability for downstream functional analysis. The next key component of the REM-I platform is the Deepcell Human Foundation Model, or HFM, which assesses 115 morphology dimensions of each cell in a sample. It does this in real time as the cells are flowing through the chip on the instrument. The HFM combines deep learning and computer vision methods to extract cell features that quantitatively and reproducibly represent cell morphology. The deep learning aspect of the HFM is based on a self-supervised learning algorithm, is trained using millions of cell images from many biological human sample types, and it detects differences in cell morphology without labeled training data, meaning that the HFM is ready to use on day one without the need for additional training. As I mentioned, one of the reasons the Deepcell HFM is so unique is that it combines both deep learning and morphometric features, providing both accuracy and interpretability. The HFM's 115 combined embedding dimensions include 64 deep learning-derived embeddings and 51 computer vision-derived morphometrics. Now let's talk about the distinction between these two encoders a little bit more. Morphometric features can be considered human interpretable quantitative metrics of cell morphology, including things like cell size, shape, texture, and intensity, as well as biological features such as blebbing and pigmentation. They also map to measurable and recognizable concepts. And shown here are some of the examples of the 51 morphometric features that we compute, starting with simple and obvious ones to ones that measure granulations and more subtle features. You can find a complete list and description of these metrics on our website in our HFM panel sheet. Deep learning features are information-rich metrics of cell morphology with powerful discriminative capabilities, but may not be human interpretable. They are quantitative descriptions of cell features derived from deep learning models based on neural networks. And of note, AI-derived features can outperform traditional morphometrics in cell classification tasks through identification on non-human interpretable cell features. Hence why we have combined deep learning with morphometrics to provide both accuracy and interpretability in a fast and effective cell feature extractor. The HFM extracts these 115 quantitative dimensions of morphology, and our software projects this data onto a morphology UMAP, where each dot represents an individual cell. Using Leiden clustering, we are able to see distinct morphotypes that cluster together in UMAP space, giving us a view of the heterogeneity within any given sample. And for a view into differences between samples, we can also examine morphology density UMAPs that show us what morphotypes are present or predominant in any given sample. This data is accessible and easy to explore in the Axon data suite that makes up the final component of the REM-I platform. Within Axon, you can organize your runs and share them with colleagues. You can look at the AI embeddings as morphology UMAPs. You can look at the images of the cells and overlay representative images from each morphotype directly on the UMAP. You can compare morphotypes across many of the measured dimensions directly on the screen. And last, but certainly not least, you can do morphology sorting, which means you can use the lasso tool to define up to 6 populations of interest for sorting into 6 collection wells on the REM-I instrument. As you can imagine, this powerful morpholomic analysis has implications for several applications across research areas. We've worked on projects in cancer research to look at tumor heterogeneity and malignant cell enrichment, in cell and gene therapy for compound and functional screening, as well as QCing cell culture, and in stem cell and developmental biology to look at iPSCs, as well as cell activation and differentiation. So, I hope now that you have an understanding of the REM-I as a tool for real-time microfluidic sorting, AI-based image analysis, and label-free bright field imaging to assess morphological signatures of cells. Thanks so much for taking the time to hear from me, and if you're interested in learning more, I encourage you to check out our website at deepcell.com or follow us on your favorite social media channel. And now for the exciting part. I'm very pleased to turn the floor over to Dr. Evelyn Lattmann to talk about her use of morpholomics in studying melanoma. Thank you.
Thank you, Michelle. As a reminder to the audience, you can submit your questions at any point during the webinar. And with that, let me introduce our speaker. Evelyn Lattmann is a research scientist at the University Hospital Zurich, and she is also affiliated with the University of Zurich. Her research focuses on cancer biology, particularly melanoma. Lattmann unravels the interactions between melanoma cells and their microenvironment, where she aims to identify potential drug targets and discover novel, non-invasive biomarkers for diagnosing and monitoring melanoma progression. Let's make sure our slides are up and running. Okay, take it away.
Yeah, hello, Charlene, Michelle, and everyone online. So first of all, thank you for the kind introduction and also the opportunity to present our collaborative project with Deepcell here. So in this webinar, I would like to shed some light on how we actually use now this morphology combined with artificial intelligence to better understand melanoma. First, a quick introduction about melanoma. Melanoma is the most invasive skin cancer with the highest risk of death. It originates from the melanocytes in the skin, which are the pigmented cells in the epidermis that produce the pigment melanin that is transferred via dendrites to the keratinocytes in order to protect them from UV-induced DNA damage. 90% of all of the melanomas are caused by exposure to UV light and by the introduction of mutations into the DNA that leads to a very high mutational burden of melanoma. Another very distinct feature of melanoma is the high potential to metastasize. Here in this cross-section, you see in situ melanoma that starts to invade vertically into the dermis. And from the dermis, the melanoma cells, they can hijack the vasculature, also the lymphatic system, to then spread to distant sites and form secondary tumor growth called metastasis. On the right, you actually see a skin section from a melanoma patient that is labeled with melanA. That is a typical marker for melanoma. You see these labeled cells here in red, that they're highly proliferative. And you can also appreciate here that this is probably a region where cells start to invade into the dermis, basically here crossing the basement membrane. Although not the most common cancer, melanoma is actually quite frequent. And in 2020, worldwide, there has been around 300 cases of melanoma diagnosed, with around 60,000 patients who died from the disease. There is an increase in incidence due to better diagnosis, but also increased sun exposure, and it is expected that in 2040 there will be around 500,000 of new cases and around 100,000 deaths. Also, in the US, the cancer is actually quite common, so it ranks among the top 10 cancers in the United States, with around 100,000 patients that are estimated to be diagnosed during this year. Approximately 1 in 50 Americans is expected to actually be diagnosed during their lifetime. And similarly, similar to the worldwide tendency increase in incidence, we also see 30 years, as we see a doubling of the incidence in the US in the last 30 years, more than a doubling actually. So here this outlines again how melanoma evolves. So basically from the sunlight exposure to the accumulation of DNA damage that then leads to the transformation of the melanocytes in the skin, in situ melanoma formation, and then finally to the spread of the melanoma cells via the lymphatics to distant organ, and for example, metastasis formation in the lung as well as in the liver. So how can we prevent melanoma formation? And so the first line of defense is actually prevention. So we can protect ourselves from sunlight, for example, using sunscreen, wearing protective clothing, or also avoiding peak sun exposure times. Then a second line of defense is, for example, regularly checking moles if there is a change in the morphology of the moles, and also we can then, if there's suspicious moles, we can remove them. And as well, removal of moles and primary tumors lead actually to a very high survival rate of patients. At the later stage, the first-line therapy of melanoma is by the use of immune checkpoint inhibitors. And these immune checkpoint inhibitors basically release the brake of the immune system so that the immune system then is targeting the melanoma cells, which is usually trying to hide from the immune system. And the use of this immune checkpoint inhibitors has really been revolutionizing the field of melanoma. So, for example, here you see a combination therapy of nivolumab and ipilimumab, which are immune checkpoint inhibitors against PD-1 and CTLA-4. And you can see that in 52% of the patients, actually, this double treatment was effective. On the downside, of course, that means also that still half of the melanoma patients actually do not profit from immunotherapy. And so these patients are then often treated by targeted therapy. In melanoma, there is an activation of the MAP kinase pathway. So a lot of melanomas actually have a mutation in the BRAF gene. that then leads to this hyperactivation of the MAP kinase pathway. And one of the inhibitor, therefore, basically inhibits this BRAF oncogenic oncogene, basically a protein produced by this oncogene. And here you can actually see that the inhibition of melanoma with this inhibitor is quite effective, actually, already in very short time. However, you can also see that there is actually a big challenge with these targeted inhibitors, that there's actually resistance also after only several weeks. So yeah, this occurrence of resistance is really a big challenge in the field of melanoma, and it is important that we can detect this the occurrence of resistance at the very earliest stages, and that we find new strategies to overcome it. Here on this slide, you see basically the evolution of resistance, and I would like to focus on the part of the different modes of how a resistance can be acquired. So, of course, the cells can already be intrinsically resistant, but they can also acquire resistance through mutation, which is then acquired resistance here labeled. But another feature which is particularly important in melanoma is actually the acquisition of resistance through phenotype switching. And since this is a very important concept in melanoma, I will highlight it a bit in more detail. So during early phase of melanoma development, the cells are very proliferative, as I've mentioned, and they have a very fast proliferation rate. So, but then due to signals from the tumor microenvironment, such as, for example, stimulation by TGF-beta, cells can switch into a more invasive state. So they actually switch to a mesenchymal phenotype, which is labeled here in red. And these cells are more invasive, so that it allows them, this phenotypic change allows them to invade into the dermis and then form metastasis. And importantly, actually, also this phenotypic switch can be reversible. So that means that then these mesenchymal cells, they can even switch back into a proliferative state and then basically outgrow again in the secondary tumor site. So at the core of this phenotype switching is actually the transcription factor MITF. MITF is driving the expression of genes that are involved in pigmentation, such as, for example, tyrosinase or also melanase. And high levels of MITF have been associated with a more melanocytic phenotype, so a more differentiated phenotype that still looks more like the melanocyte cells, actually. And these cells are shown to be more proliferative. However, then, as I mentioned, due to external signals such as, for example, TGF-beta or also receptor tyrosine kinase signaling activation, these cells can switch into the invasive phenotype and then activate, for example, EMT, so epithelial to mesenchymal transcription factor, that leads then to pro-invasive gene expression. And so these cells then are characterized by low levels of MITF, but rather high levels, for example, of this receptor tyrosine kinases and, for example, AXL. Importantly also to mention is that very high levels of MITF or absent of MITF then lead to complete senescence. You can also see here actually on this slide that this shift in MITF is gradual, so there's also cells that kind of form an intermediate phenotype. And so just to remind you again, it's really like this phenotype switching that is actually in melanoma cells often at the core of developing resistance, because you can imagine, for example, if the cells are in a proliferative phenotype and the drug is given at this stage, they can just switch into another cell type, avoid, for example, certain signaling pathways, and like this become resistant to the drugs. And because of this important role of phenotype switching in shaping the melanoma biology, but also resistance, we wanted actually to find a way of how we can detect these phenotypes in an unbiased and also label-free manner. And of course, that's why we then focused on morphological differences, because what we actually can see here is that melanocytic and mesenchymal cells they have also different morphology as a consequence of this phenotype switching. So the melanocytic cells, they have a more melanocyte phenotype with like dendrites, whereas the mesenchymal cells, upon basically undergoing this invasive switch, they look more like fibroblasts. So they're flatter and have less of these dendrites. So then since we wanted to profile basically morphology, we teamed up with Deepcell, who is really like at the forefront of this morphological profiling using artificial intelligence. And our goal was to, as mentioned before, was to develop a classifier that can distinguish between the proliferative and invasive phenotypes in melanoma. So this brings me to the outline of the study. So in the first part of the study, we developed the classifier. So basically, as an input, we had melanoma cell lines that were derived from tumors from melanoma patients that were treated at the University Hospital in Zurich. So we classified them by RNA sequencing into melanocytic and mesenchymal cells in order to have a baseline for or a reference for our classifier. We then actually introduced these cell lines these cells into the system that is coupled to the imaging device. We then ran the classifier that was, as mentioned, based on morphological features, deep learning features, as well as morphometric features. And we then, based on this classifier, we then predicted the phenotypes of mesenchymal versus melanocytic cells and compared them with the ground truth. So then in the second part of the approach, we then went on to actually now validate our predictions with patient-derived cells, with actually cells derived from tumor samples that we dissociated directly from the tumors, as well with cells that we perturbed chemically or also had genetic mutations. So we followed for the validation experiment exactly the same procedure. We introduced them in the system, ran the classifier, and made the predictions. And we could then also do a dimensionality reduction to then displace basically the different morphological phenotypes on a UMAP. Also, in this case, again, the baseline was by RNA sequencing. So we employed a single-cell RNA sequencing to basically classify into melanocytic and mesenchymal cells. And so this brings me to the first results slide. So where we basically, where you can see like now a morphological UMAP that was where we also use Leiden clustering then to show the different types of morphological or morphotypes actually. And here you can see that for each of the morphotypes that we also have like a visual representation. And interestingly, you can see that from these 19 patient-derived cell lines, we get 10 different morphotypes. You can see actually one morphotype is quite distinct from the rest, and this contains the pigmented cells. You can see here that actually these cells appear to be darker because they contain the pigment melanin. But also within this cluster of pigmented cells, we can actually split them again into two populations that have a different appearance that is captured by our classifier. But we were interested now, of course, in like this different, in actually distinguishing the melanocytic and the mesenchymal phenotypes. So we were wondering if we now map actually the phenotype of these cells onto this morphological UMAP, whether we can get a distinction of these two different cell states. And indeed, this is actually what we see. You can see here again on this morphometric UMAP here on the left, as well as on this morphometric UMAP on the right, is that actually the melanocytic cells, they populate different places on this morphological UMAP than the mesenchymal cells. So they're almost mutually exclusive. So that really means that we actually have two different morphotypes that define these cell states. In the middle here, you actually see now that this is also consistent across cell lines. So for this, you see 3 representative cell lines where you can see that actually these melanocytic cells, they populate the more lower left of the morphology UMAP, whereas the mesenchymal cell here on the right populate more the upper right side of the morphological UMAP. And again, you have here also the visual representations. And I would like to draw your attention again here on this like middle cell line of the melanocytic cell lines, where you can actually see that we have this also the occurrence of pigmented cells, which we see in this very distinct cluster. So this was very exciting, and so that's why we wanted to see how our performance, how our classifier actually performs. And here you can see this in the confusion matrix, that we had actually a very high accuracy of the classifier. So in 88% of the cases, melanocytic cells were classified correctly. In the mesenchymal cell, this percentage was at 83%. So we had a very good performance of this classifier. And so we wanted to know which features are actually responsible for this outcome. And so we distinguished between the deep learning features as well as the morphometric features. And you can see here in these graphs that actually the deep learning features are better in distinguishing these two phenotypes. And for the morphometric features, if we look at the ones that are performing the best, we see here LBP periphery 6. So that's actually referring to the local binary pattern, which gives us an indication sort of like the granularity of the cells. You can see here, this is showing what this feature is showing. It's kind of like how the granules in the cells are organized, and which is recognized by this feature. So if we then look at overall at the classification of the cell lines, we can actually see that in all of this 19 cell lines, we get the correct we get the correct classification. So all of the melanocytic are labeled as melanocytic, and the percentages range from like a 77% to like 97%. And I think that also makes sense that we don't have like 100% of the cells labeled as melanocytic, because these cells, they still have a little bit of heterogeneity. So not all of the cells may be actually melanocytic. So that also makes biologically sense. So next, we went on to validate our classifier. And as I mentioned, we used perturbation experiments to do so. So we had cell lines, control cell lines that we then exposed to stimuli. We introduced both of these cells into the system and then ran our classifier and recorded the predicted percentages of melanocytic and mesenchymal cells in the control samples, as well in the perturbed sample. And we compared the two to see whether we see now a shift in phenotypes that we can predict with our baseline, which was again a single cell RNA sequencing. And so to hear an example of the first perturbation that we performed. So the first perturbation was the use of PLX, which is a vemurafenib that I introduced you before. This is a BRAF inhibitor that targets the oncogene BRAF. And this leads actually to a shift from a more mesenchymal phenotype to a melanocytic phenotype. And this is actually exactly what we see if we use our classifier. We can see here again on this morphological UMAP, you see the control cells here in purple. They populate the more mesenchymal part of the UMAP, which you can see here again, the reference, which is in the upper right. And then upon exposure to vemurafenib, the BRAF inhibitor, these cells, they undergo a shift towards a more mesenchymal phenotype. You can see here the corresponding pictures, images that for the control as well as for the vemurafenib treated melanoma cells. We can also quantify these results. Here you see again that we have like this shift from a mesenchymal phenotype to a more melanocytic phenotype upon treatment that we confirmed by RNA sequencing. So we then went on to further perturb the cells. We added, for example, again, vemurafenib inhibitor, but now to a different cell line, the SKML28. And similarly to the A375 cell line, we see again the cells, they start out as more melanocytic, but then turn into a more mesenchymal phenotype. And you see again the images below here in the control as well in the perturbed situation. But then we went further also to try different types of perturbations. So I've mentioned to you before that, for example, TGF-beta is introducing an epithelial to mesenchymal transition in melanoma cells, which is basically a shift from a melanocytic towards the mesenchymal phenotype. And this is what you can see here also in the morphometrics, is that we now, that actually these control cells, they're shifting from a more melanocytic phenotype to a more mesenchymal phenotype here on the top right. So since this worked quite well, we went on also to test another perturbation, which was actually a single mutation in a gene, STK11, that is an inhibitor of invasion. And this STK11 was knocked down by CRISPR previously and has been shown to actually make cells more invasive. And if we now compare this STK11 CRISPR knockout cell line to our control cell line, we see that again we have a shift. So like now the opposite shift, basically from a more melanocytic phenotype to an invasive phenotype, which is consistent with what we expected, and also confirmed by RNA sequencing, single-cell RNA sequencing. Then last but not least, we also had basically a natural occurrence of this phenotypic shift in cell lines from one patient. So this patient, these patient-derived cell lines have exactly the same genetic makeup, but they have a different phenotype. And we imaged both of these cell lines, and again, we could see that actually different phenotypes, like so the upper cell line is a more mesenchymal cell line, so which we could correctly see on this on this morphological UMAP. And here again you have the pictures showing the different visualizations. Next, we wanted then to move on to patient-derived material, and for this we had 6 tumor samples, melanoma tumors, that we dissociated into single cells. These single cells we then again input into our system. We imaged them and ran our classifier and made predictions for melanocytic versus mesenchymal cells. We then also made again this morphological MUMAP to see basically the different morphotypes. And as a ground truth, also again in this case, we used the single-cell RNA sequencing. And here is just like the overview of now the single-cell RNA sequencing UMAP. You see here like that we can get a nice separation also of the melanocytic cells, which are here in the lower left, and also the mesenchymal cells here on more the upper right. So this so RNA sequencing was kind of successfully generating our baseline. And here below, just like as a reminder, you see again the morphological UMAP. So now when we look at this clinical, so yes, one thing I have to mention also, so not to introduce any any bias, we actually we just went for tumors. So we went for dissociated tumor cells that came from tumors that had more than 80% of tumor cells, but we didn't introduce any sorting. So we just took all of the cells without doing any pre-sorting. So we most likely also had some stromal and fibroblast cells in our preparations. And so, yeah, here you see the outcome of this cell sorting. So again, we have like the morphological UMAPs where we can now map the dissociated tumor cells onto it. And you can see here below now, in contrast, the single-cell RNA sequencing UMAP. And here again, actually, interestingly, what you can see is here in this third dissociated tumor cell sample, we see again actually the pigmented cells that really come up in this, from our classifier. And overall, what you can see here is actually that we see a rather melanocytic phenotype in these DTCs. And maybe this is now better to go to the next slide, because you can see here also the quantification. So this actually also corresponds to the single-cell RNA sequencing data, so that actually most of the DTCs, except for DTC4 here, is characterized as melanocytic. And There is also actually one outlier here that I have to mention. So for example, like in DTC2, we didn't get the right prediction, so we don't exactly know why this is the case. It could be, for example, that this is from another organ. So this DTC is not from lymph node, but from the skin. And our classifier was mainly trained by lymph node samples. So this might be at the origin of this discrepancy. But overall, we see that we are able to predict the right phenotypes also in DTC samples. And this brings me basically to the conclusion. So high-dimensional morphology analysis reveals multiple morphotypes of melanocytic and mesenchymal cells. Then these morphological differences were used to develop a melanoma phenotype classifier that can predict phenotypes with more than 80% of accuracy. Then we can actually use now this classifier to predict the phenotype also of dissociated melanoma biopsies from melanoma patients. And we can, so this allows us then to really characterize cells phenotypes in a label-free manner, and this can give us more new insights into tumor biology. And I think it's also important to point out for the future, this label-free isolation of cells will allow us to explore more diverse behavior, for example, in drug screens, and it will also allow us to detect distinct states in liquid biopsies eventually. And so with this, I would like to acknowledge everyone who has contributed. So it has been a very nice collaboration with Deepcell. It was great that we could use their state-to-the-art technology and the know-how they provided. So in particular, I would like to thank Andrea, also Julie, and Manisha, and everybody who is listed here in the team who has helped really driving this project forward in a very successful manner. At the University of Zurich, I would like to thank Mitch, who is running the melanoma lab that I'm working, and he has several exciting and impactful melanoma research projects going on. I would also like to thank Aishan, who is an expert and great in single-cell technologies and a great lab colleague and collaborator. And last but not least, I would like to also thank the patients of University Hospital of Zurich that have provided us basically with their samples. And without the patients, of course, this study wouldn't have been possible. And so, yeah, thank you also for listening. And I'm happy to take also questions.
Thank you for that presentation. The audience has submitted several questions, so let's get to them. Can you perform mixed cell population evaluations without being able to run a classification training set?
I'm happy to take that question. So the HFM is actually integrated into the REM-I, so you can put any sample on it and get data out. The HFM will identify those differences between cells in a sample based on that 115 dimensions, and then it will project those predicted morphotypes onto a morphology UMAP. So yes, you can perform mixed cell population evaluations.
And how would you validate such an experimental model?
So to validate something like that, you'd probably want to go ahead and sort those cells and confirm with an orthogonal data type. So there's kind of like two ways going into that. You can, confirm them up front and then put them in separately and create that classifier, or you can just do more exploratory by putting it in, let the HFM evaluate what's there and then confirm afterward, because it does sort and keeps those cells viable.
Great. For Evelyn, now that you've developed your melanoma phenotype classifier, how do you plan on using it?
Yes, I think there's several ways of how we can use it. So I think one line where we like to use it is, of course, like then for testing, for example, resistance, as I mentioned. So we can actually now separate these lines from tumors in a label-free manner. And I think this will allow us then to also in vitro test different drugs. I think it's also one thing that we would like to do also with this classifier, would like to further refine it so that we can also actually capture these intermediate states. For the moment, we just have like basically use the binary classification, so melanocytic and mesenchymal, and we did not focus on the intermediate states. But I think this will also be very interesting to expand there, that we can also get these intermediate cell populations and then basically make predictions of how they. So basically to isolate also these, and then see basically if we isolate different morphotypes from tumors, how do they respond to different treatments, and how that also relates then to, for example, RNA sequencing data. I think also things that are interesting to look, for example, is like we have seen that with a single mutation we can shift the morphology phenotype. So now this of course also opens possibilities by basically performing CRISPR manipulations and tend to check, for example, for the morphology that is linked to this particular gene.
Wonderful. And for both Michelle and Evelyn, how do you foresee researchers using Deepcell's REM-I platform in the future? Do you want to start, Evelyn?
Yes, sure. Yes. Yeah, I think that there's a lot of a lot of possibility. I guess it depends on what you're working with. I think in cancer, I think as mentioned, I think for resistance and also eventually probably for actually detecting cells in liquid biopsies, I think that's something that could be very interesting if we then can actually detect, for example, melanoma cells in the blood, and if we can actually also phenotype them in the blood, or for example for blood cancers where we can maybe see different phenotypes already directly within the blood. But then I think also other areas will be interesting, for example, like if one can look for different morphology of blood cells. For example, sickle cell anemia gives different morphologies of red blood cells. So I think this technology could be useful to identify these cells, or just like bacterial populations where you want to see maybe resistance to certain antibiotics, or I guess also like cell biological questions. I think maybe one could even go down to also sort organelles or that are released by cells. So for the moment we have focused on cells, but of course I think with a better resolution maybe one could also look at things that are actually secreted by cells.
And Michelle?
Yeah, I agree with Evelyn on a lot of that. I kind of think about the REM-I and its application at kind of a really high level. There's kind of three main use cases that stand out to me. First is using morphology as a biomarker, so being able to, predict cancer or other diseases as kind of a marker itself. I also see it being used for evaluating response to perturbations, like Evelyn showed, or in things like CAR T development. And then kind of lastly, the REM-I is really good at evaluating kind of the really subtle differences on the continuum of differentiation. So we also see a lot of interest in various stem cell applications. But I think the applications overall are very varied and applicable to a lot of different research areas.
Great. So as a reminder to the audience, the recording will be available after the presentation. It'll be sent to you within a couple days. Another question for Michelle. If right now, in order to study morphology, most people will use either microscopy or flow cytometry. Can you compare and contrast those techniques with the REM-I?
Yeah, that's an excellent question. So the beauty of the REM-I is that it kind of brings the best of all of those together into one platform. So with microscopy, you're able to, look at cells. You might even be able to, take the image of them and do some manual analysis on it or into some imaging software, but it's pretty low throughput. Flow cytometry has the value of being extremely high throughput, but often relying on labels. And so, and not necessarily having that high dimensionality of the AI that we have. And so, and then, depending on the tool you're using, if you want to actually keep those cells viable and sort them and do additional analyses on them, that's really where the REM-I shines, right? It's able to kind of bring all those three things together into one platform and then also provide that sorting.
And now we have time for just one more question. What's the throughput efficiency if you were to use the REM-I for isolating a target cell population label-free?
That is a very good question. If I'm interpreting throughput efficiency correctly, it kind of, as more like purity versus yield, it's actually kind of a tunable parameter for us. So you can run the instrument very quickly. If you want to, say, do downstream analysis for like bulk sequencing applications or bulk RNA or CNV analysis. And then you can actually slow it down if you really care about capturing really, really high purity in order to be able to do like single cell RNA-seq. I apologize, I don't actually know what the exact numbers for each of those right now because it's a tunable parameter, but we're developing a graph that will show that. And so keep looking at our content for that one.
Excellent. Well, thank you, Evelyn and Michelle, for that discussion. We appreciate your presence here today. If anyone has further questions, please consider reaching out to the speakers directly. Their contact is shown on the screen. As a reminder, the webinar will be archived on the scientist website, and you'll receive an email notifying you when the on-demand webinar is available. Make sure to follow the scientists on social media for updates on future webinars. I would like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our speaker, Evelyn Lattmann, as well as our sponsor, Deepcell. Thanks, everyone, and goodbye.